Hello, my name is Josh Neufeld, and I'm a professor in the Department of Biology at the University of Waterloo, and it's a pleasure to be speaking with you today about the microbiology of barrier components of a deep geological repository for used nuclear fuel. And this research direction is part of a broader portfolio of research that spans discovery-based research, like nitrogen cycling organisms, or the discovery of novel taxa and their functions, all the way to applied aspects of that research, like applying those organisms to better treat water and engineered aquatic environments, for example. And in the last 10 years, we've focused increasingly on the management of used nuclear fuel and the microbiology of a deep geological repository. And that's because we partnered with the Nuclear Waste Management Organization, the NWMO, which was charged to identify a solution for Canada's high-level used nuclear fuel. And so the funding for the NWMO comes from the nuclear energy companies in particular. And so according to the polluter pays principle, it's important for those companies to be responsible for the management of the waste associated with that energy. So that's what we do, and it's important, especially for the University of Waterloo here. We're within the province of Ontario, and most of Ontario's energy comes from nuclear. And of the 4 active operating power plants in Canada, 3 of them are in Ontario, and this is the smallest of them, the Pickering nuclear power plant, and it has 8 reactors, as you can see right here. The reactors have fuel in them that lasts in the reactors for about a year to 2 years, and then the fuel needs to be stored. It's spent, it needs to be stored, and it needs to be stored long term. What does the fuel look like? I'll just give you an image of it here. I even have what is the same a cylinder the same size as a fuel pellet, and this would be this is uranium dioxide pellet, and it's a ceramic, so it's hard. It's not a liquid, and this fuel, this uranium dioxide fuel, is placed, these pellets, inside of these fuel pencils. This is zircaloy tubes, and the tubes are welded into a fuel bundle. The bundle is about the size of a fire log, but it's a lot heavier. It's about 50 pounds for each bundle. The bundles stay in the reactors for a year or 2, and after that they need to be stored. They are quite hot when they come out of the reactor. They're highly radioactive. They generate heat, and so they need to be cooled. And so they are cooled for about 10 years in these pools. And then at that point, 99% or so of the radiation is now decayed, and they are cooler, and they can go into dry storage containers as shown here. And they can stay there indefinitely, but the containers have a lifespan, a lifetime of several decades. And even the buildings that are hosting these containers have a lifespan of a few hundred years, and they are located on the sites of the reactors themselves, and these are typically on shorelines. And so this is not a long-term sustainable solution, because there's going to be a couple of ice ages, and the ice ages will then have kilometers of ice above all of our heads. And so it's important to have a long-term sustainable solution to allow the used fuel to return the amount of radiation back to background levels. How much are we talking about? In terms of used fuel, these bundles, there's about just over 3 million of these bundles that are being stored currently. And by the time a long-term solution, a deep geological repository, is implemented, it may be that there's twice that many. But just over 3 million bundles would fill, just as for an idea, these 9 hockey rinks from the ice up to the boards, and that gives you an idea of the scale of the used fuel problem that needs a permanent long-term solution. There are As part of the design of a deep geological repository, the plans include a variety of barrier systems, engineered barrier systems and natural barrier. The engineered barrier systems start with the zircaloy ceramic pellets themselves, which are resistant to release of the radioactive material, so that itself is a barrier. The next is the zircaloy tubes that are in the fuel bundles. Those are resistant to corrosion as well, and that is then complemented by the used fuel containers. The design of these is really convenient with the carbon steel containers coated with 3 millimeters of copper, and that copper coating provides corrosion resistance. And then there's a bentonite layer, bentonite clay, which I'll talk about soon. This bentonite material is both powdered bentonite that makes up these boxes that surround the used fuel containers, and then there's the gap fill bentonite that's blown in. It's a milled powder, granulated form of bentonite that can be blown in and surround all the spaces that surround these buffer boxes. And then there's the natural barrier system, the deep subsurface, and we'll talk about that natural barrier towards the end of the presentation. The subsurface, just to give you a little idea of why the subsurface may be ideal for a deep geological repository, is because with increasing depth in the subsurface, there are fewer and fewer fractures that lead to very small amounts of less and less water and space for microorganisms that would primarily be contributing to the corrosion of some of the natural barrier components. So relatively few fractures, you have increasing temperatures with depth, you have increasing pressures with depth, and because of a combination of characteristics, the diversity and the abundance of microorganisms tend to decrease with depth. It's really challenging to obtain enough energy, nutrient, space, and water as you go down in the subsurface. And I'm not going to get into too much detail here, but there is potential for microbial life in the surface, and for sure that is because of the availability of different energy sources and carbon sources, perhaps more than would be expected, that you might consider. And so one of them would be hydrogen gas. This is a source of electrons and energy, that this is produced slowly from radiolysis, from rock itself, and so that's one source of this electron donor, which can be coupled to a variety of different electron acceptors, which means there's potential for microbial metabolism. You can also have energy from necromass, like dead cells and so on, and other organic sources of carbon that can be used as a carbon source, as an energy source. And so that can be coupled with different electron acceptors as well for different respiration processes and so on. So what's of concern here for the deep geological repository? In particular, it's this coupling of different electron acceptors with sulfate as an electron acceptor, because it's the sulfide, this rotten egg smell hydrogen sulfide gas that's produced, that can have the potential to directly interact with the used fuel container. In this case, that corrosion resistant copper layer can be corroded by sulfide if it's present and produced in the adjacent subsurface, and so that could compromise the integrity of a used fuel container. So it's the sulfate-reducing bacteria, the SRB, that have been traditionally quite a focus of microbiological characterization in a nuclear waste context. There are other metabolisms that are of interest as well, like denitrification, for example, or methane generation, methanogenesis, fermentation. Anything that produces gases as an end product potentially could contribute those gases that could generate permeability fissures to allow in water or gases into a DGR. And so that's why all microbial processes are considered together when exploring the suitability of the conditions proposed for DGR and the incompatibility with microorganisms. So that's why we have a multi-pronged research effort in my lab that focuses on those engineered and natural barrier components, including the bentonite clay that goes into the buffer boxes and the gap fill. We have pressure cells that contain clay that we incubate under DGR-like conditions. And we also characterize subsurface samples from that are relevant to a DGR. We do that with cultivation-based approaches, those traditional approaches that can be biased, but we complement those with the molecular profile data as well that provide a more complete picture of what is potentially there in our samples. And we do metagenomics as well, not just single gene analysis, but we do metagenomic analysis. But I'll be focusing today on the cultivation and ribosomal RNA gene analysis to show you what's found in the bentonite, in the pressure vessels, and in the subsurface. I'll focus on the bentonite first. This is a really important subsurface component here for the engineered barrier components. And the reason why the bentonite is so important and what the characteristic that makes it so useful is its swelling ability. If the bentonite that's shown in this tube here is wet, so you can see I've just added some water, and you can see right away, and this is a stop motion where I've taken pictures every so often and made a video out of it, you can just see how the bentonite swells. The bentonite naturally absorbs because of the montmorillonite clay that's part of this benton deposit from Wyoming material. It pulls in the water into the structure of the clay. And yes, it swells. It swells, but, and I have some of the clay material right here that's been packed nice and tight. If it's packed nice and tight like this and kept from swelling, from expanding, then it will develop very high pressures, so it won't be allowed to expand. And the high pressures result in very small pore sizes, which then have very low amounts of water, and all of that is really helps to prevent microbial growth. That's some of the features of this clay that are so useful for use in a potential DGR. Okay, so what we wanted to know is different clays, what types of microbes come in with the clay? What do they contain? And so we explored clays that are intended for a DGR, that could be used in DGR from that Wyoming deposit. So all of these MX clays that you can see right here, they're for MX80. They're for a sort of referring to the size of the powder of the clay. These all are from Wyoming, and then we have other international clays that are similar with high montmorillonite, that swelling clay that offers those properties. And we compared them all. And when we're looking at molecular profiles here, if you look at a high level, like at the phylum level or the subdivision level here, you end up with very similar overall profiles. And they look similar, but they're actually not. If you look at the ASV level, so the individual taxa, there are differences between the clay, and they're sort of predictable based on where the clay came from and when the clay was mined and processed. And so you can then take all of those profiles at that high taxonomic level and compare them in ordination space. And so in this 2 dimensions here, you can see samples that are close together are more similar to one another based on their 16S ribosomal RNA gene profiles. And they tend to be similar in terms of lot number when they were processed and in terms of their location, like MX80s versus other international bentonite sources. So that means that they have their own microbial signatures. But if you were to culture all of these clays, as we did, on different growth media, under different conditions, here's where we don't see so much diversity. We end up seeing the same things, and even here it's a bacillus that we tend to grow under aerobic or denitrifying enrichment conditions. And then we have these sulfate-reducing bacterial enrichments here. So these are all mostly desulfosporicines species that we grow out of all of the bentonites. So there's a lot of commonality in what can be cultured from the clay. So despite those unique signatures that we tend to obtain, you end up with very specific taxa that we can cultivate. And that's that you can see here at that ASB level. So at the highest taxonomic resolution possible, you can see, if you summarize all the data, you have some things that are very abundant in the dry clay, that if you just extract the DNA from the clay and sequence it, some things can be quite abundant, but not cultured. And other things are very abundant in our cultures, these black circles up here, but they're not seen in the clay profiles themselves. So maybe we didn't sequence deeply enough. And then there are other things that we can culture and we also see in the clay. So everything in red we can culture and see in the clay. So the bacillus we see in the clay and we can culture it. The streptomyces, very high in the dry clay, we can also culture it. So that shows some commonality, but a lot of things you don't see with one method or the other. One of the advantages of using two methods like this. That's what's in the dry clay, as received clay. What happens when you wet the clay, which will happen in a repository? It will get wet, it will saturate, and it will have different amounts of water allowed into the clay depending on how much clay is packed in before that saturation happens. So we simulate that in the lab where we add a small amount of water, very small amount. We add a medium amount that's a little bit more in line with what the repository will have in terms of water activity. And then we add more water, which would be high. This is higher than you'd expect in a repository. So we compared that. What grows in the clay? And these are all the same images here, just showing you that we put put all these different clays into mason jars and incubate for different amounts of time. And we published these results, so if you'd like to see them, they just came out this year. This is Rhiannon Punch's work, a master's student that finished in my lab. And what I'm just showing you here, it's a collaborative project with other co-authors, but in terms of the actual microbiology data we generated, I'm showing you here as received clay with gray profiles over 6 months. This is all oxic conditions, so in the presence of oxygen. And if we hydrate it with water, you can see the different dry densities. You see an increase here in the ribosomal RNA genes we can extract and amplify. And that's true also for the aerobic heterotrophs that we grow on these R2A plates. Here we see an increase, especially with the high water activity. So if you add more water, we really do see a greater increase in the abundance of what's growing. But there's some aerobic heterotrophs growing in at all time points. That's not true for the anaerobic heterotrophs so much. You don't really see those in the water. You might wonder what CR10 is. This is a simulated groundwater, which would be a little bit more realistic, not just water, but there's some salts in it that represent what a groundwater composition might be. So you sort of see a similar effect here, an increase in culture or in templates from the DNA. But we don't see anaerobic heterotrophs. This is a worst case scenario. We use just straight up sulfate-reducing bacterial medium, growth medium. So there's lots of organic carbon here for anything to grow on. And so you have the highest increases we saw in the 16S ribosomal RNA genes. Aerobic heterotrophs go up, and even the anaerobic heterotrophs increase here. We don't see, interestingly, sulfate-reducing bacteria at all, but these are oxic conditions. So we need to compare to anoxic conditions. And by the way, these red arrows are just the limit of what we can confidently quantify using our cultivation-based approaches. Then we have anoxic conditions, and here we don't really see growth at all. So you wet the clay and you don't see anything growing. So it looks like oxygen-based metabolism is really important to see growth in the clay when wet. A DGR is anticipated to very quickly go anoxic, and so these results are actually quite encouraging to see that the clay doesn't foster and host the potential for much microbial growth, even without the pressure that would be associated with the deep geological repository. And that's really what I wanted to talk about next, is what happens if you add pressure to these hydrated, saturated bentonites. So now just add the pressure. So that's where we use the pressure cells for that, for those experiments. And the pressure vessels can be put below ground in the subsurface to simulate an actual DGR, like we do at the Grimsel Underground Research Laboratory in Switzerland. And so this is a multinational consortium with different nuclear waste agencies, all exploring what happens to the bentonite and also the metal coupons that are placed within the bentonite in the subsurface. So now we're under higher pressure. And so that's what it looks like. I mentioned the metal coupons that are placed within the bentonite here and incubated in the subsurface. And so collaborators are exploring the metals to see how much corrosion is associated with them. And my lab is exploring the microbiology of the clay in the subsurface under DGR-like conditions. And I'm going to show you results from the one-year paper that we published in 2019 here. But they're consistent largely with what we saw in the 5-year and the 7-year. And now we're looking at the 9-year as well. So we have a number of different modules retrieved at different times and then are taken apart and carefully sectioned so we can explore the inner layer and the outer layer that's in contact with the fluid in the subsurface. So that's the results that we see here with cultivation-based approaches. Everything we look at here, I'm showing you bubble plots here again, which is just showing you taxa and the relative abundances. And you're looking at here bentonite inside, bentonite outside, the very outside of the pressure vessels. And you can compare that to the powder and the pellets on the right side here that went into the pressure vessels in the first place. They went in at 2 different dry densities, meaning you pack in 1.25 grams of dry bentonite per centimeter cubed or 1.5 grams per centimeter cubed. And 1.5 is closer to what's anticipated to be the amount of bentonite you pack into a deep geological repository. So that's closer to repository conditions than the lower one is. So the lower really offers a comparison for what if the conditions are variable and you have some regions with lower dry densities. Anyway, what we do see is that the powder and pellet profiles look a lot like the inside bentonite profiles after a year of incubation, wet and under pressure. Here we're not seeing much change at all. You have very similar profiles. The outside of the bentonite also looks a lot like the original powder and pellets, but here is where you start to pick up some of the taxa that we see in the transport container. So it's chicken or egg where this came from, whether it's coming from the outside of the bentonite or the transport container is associated with that very outer layer and sort of infiltrating that. But these profiles are all completely distinct here from the borehole fluid. So what's found in that subsurface rock that's now the water that's hosting these pressure vessels and it open to permeating to the bentonite itself. Here you're seeing very different profiles. And the profiles do have sulfate-reducing bacteria in them. And so these SRB here, desulfosporosinus, we often see. There's a relatively high amount of them, but we don't see those at all in any of the bentonite. So they don't seem to pass into the bentonite, and they certainly don't make it to the inside near the metal coupons, which is really promising that there's unlikely to be infiltration of organisms directly to the metal of the used fuel containers, as provided the bentonite barrier is in place. And we also don't see very much difference between the 2 dry densities, so there may be some flexibility in the targeted dry densities in the end. That's a 1-year snapshot time point. We also do this in the lab with pressure vessels that are set up under different conditions. We can vary more conditions and test them, but at least from the subsurface data, we can say that there is evidence that there's stability of microorganisms in bentonite under conditions like a DGR, and then we look to the lab to explore shorter time frames, different conditions. And so that's what this paper we just published last year shows, that that if we were to compare the powdered bentonite to gap-filled bentonite at different dry densities, and then explore the clay inside these pressure vessels after different time points, what do we see? And so I'll show a lot of the data all together in this one multi-panel graph, which has ribosomal RNA genes in purple, aerobic heterotrophs in blue, anaerobic heterotrophs, and then SRB in green. And you can look here again for reference, for comparison. Look at the gap fill, no hydration, just straight up gap fill material, or powdered bentonite material, relatively low abundances of everything. And then you wet it in the pressure vessels. You allow them to saturate. It takes time, but you push water into the vessels and you allow them to saturate. If you only put a relatively lower dry density bentonite in there, they saturate very quickly. And so that's true, the 1.25 powdered, the 1.1 powdered. You can sort of see here with these water activity plots that when you have higher dry densities, it takes longer for the water to get in. And some of them don't fully saturate during the whole 12 months of this incubation that I'm showing here. But when you look at the data, what I find quite interesting is that even at 1 month, you do see an increase in 16S and what you can culture out of the clay. And if the dry density is sufficiently high, that goes back down within that year or 18 months or 6 months, whatever it is. It goes back down to background. So there's a temporary increase in abundance of what you can culture, and then the 16S goes up and actually stays up, even though the culture drops back down. So that's true for the inner layer samples, but it's true for the outer layer. So that's happening on the inner. What that implies is that as the water is infiltrating the pressure vessel, that there's a transient saturation front that's associated with incomplete saturation, and the pressure has not yet been fully realized within the vessel. And so you can get growth as the water is moving through the bentonite, but that once the bentonite is saturated and the pressure has reached its fullest potential, that's where you see a decrease in what can be cultured out of it. So that implies that the viability of the organisms in the clay, even though they may temporarily go up, quickly return to background levels, provided you have sufficient dry density. So, and these results right here are all under oxic conditions, so that they're not. This is not anoxic. So we have data that we're generating that are related to the anoxic conditions that I'm not going to show today for the interest of time. But this just gives you an idea of what happens when you're just saturating under sort of worst case in terms of oxygen conditions. But it's also important to notice the green bars, the sulfate-reducing bacteria, they don't increase significantly above background in any case. So that's, again, consistent with what we have seen in our other incubations. SRB don't grow in bentonite clay under most conditions. We also did the profiling on the DNA level of all of these pressure vessels, and there's too much to go through here, but one of the take-home messages is that when we see, we saw the growth associated with that saturation front, it's the Pseudomonas that you're seeing here that really seems to increase and stay within the profiles for most of the time points. They seem to be decreasing at the 12-month time point here under the higher dry densities. They go back down quite a bit. But it's the Pseudomonas that tend to be proliferating. So we are focusing on culturing and characterizing this Pseudomonas to identify what they contribute as they grow in the saturation front. And the bacillus as well. There seems to be some bacillus increase. So that's what's happening in those oxic pressure vessels. Then lastly, I just wanted to share some of the preliminary data that we have based on the subsurface, and part of the reason why they're preliminary is because there has been a site selection process that's ongoing, and that has completed. So the selection of the host site has now been announced that the Wabigoon Lake Ojibwe Nation in the Ignace area in Northern Ontario is the willing and informed community that has stepped forward asking to continue to be considered as the host site for this repository. And in some ways, this may be an ideal location, in particular because of its crystalline rock formation in the Canadian Shield, so very stable, long-term potential location with very low amounts of water in the subsurface, so very, very fine fractures and very few of them. So we have begun to characterize the microbiology of that subsurface at that site. And so I'll just give you a snapshot of some of the results that we have generated thus far from the site. And so characterizing both the water, as this is showing you here, sampling of water from the site, and also sampling of rock core material that will then be, for us, crushed and into powder that can then be, DNA can be extracted from for those endolithic inside of rock microorganisms. So we use a variety of approaches like microscopy, but also the DNA approaches that you've already looked at, and metagenomics as well. And that's complemented by PLFA, phospholipid fatty acid, biomarker analysis by Greg Slater's lab at McMaster University. And this is all focusing on that site. It's often referred to here as the Revel site because of the Revel batholith, referring to the geological formation, and that the DGR is proposed for. And some of the work that we've did initially on some samples from the site were already published a few years ago. And that's what I'd like to just show you, sort of like the preliminary data of both the rock core and the water to provide an idea of what we're anticipating finding as more detailed site characterization continues. So we have 3 rock core samples that were ground and powdered, and so we have them from different depths above repository and then about 500 meters approaching repository. The depth that's proposed is I think is at the 750 meter level at the moment, and we have some very shallow groundwater to show you some results from as well. But that's not repository. The Revel rock has been very difficult to work with, and the biomass is really undetectable as the take-home message here. It looks from these profiles here, from these 3 rock cores, both the inner core that's powdered and extracted and the outer that we have a sample from as well for comparison. It looks like there's a lot there, but there really isn't. And in fact, these profiles here we have no confidence in because they are not consistent with each other. There are almost no shared taxa between all of these samples. So this appears to be stochastic, sort of so the random compilation of contaminant organisms, the kind of data we would generate from our controls, our reagent controls. And so we have no confidence that these are actual subsurface profiles. They could just be contaminants. And so they're not meaningful, which is good news in a way that whatever biomass is present within the rock, at least with the methods we've brought on these initial samples, are insufficient to detect anything. So that's in a way good news, that the very low microbial profiles in the rock itself. And that's also seen in the quantitative PCR data. So we quantitatively try to count templates. And so although it looks like there are signals coming from the samples that we're quantifying here, they're at or below the levels that we see in our no template controls, our kit controls. So we have no confidence we're detecting anything in the rock. So very, very low in the rock. We continue, though, to propose analyzing rock core samples from the site to ensure that the data remain consistent and remain below background levels. So that's part of our ongoing characterization. The water, in contrast, can be filtered, so in a way concentrating the signal, and we always include many different reference samples. So we can include all the drilling water samples that are collected during the borehole drilling process, and throughout the process we're collecting multiple blanks that are treated like samples in the field, but have nothing in them, and kit controls and no template controls. So all of these different controls that help us to interpret our data. The drilling water produces consistent signals, and we're confident in those profiles, so they're not as low biomass. All of the controls you're looking at on the right side are generating, as you can see here, profiles that are inconsistent with one another and are typical for these types of negative controls in environmental microbiome studies with very, very low biomass samples. And then we have our groundwater samples. So the only sample here that's actually relevant to the actual groundwater is this GW001. And so we show that next to the drilling water samples and the controls to show that what we see in the groundwater is distinct from all of those. And when you zoom in on just the groundwater sample, you see that there actually are a lot of different types of bacteria and archaea found within that one sample. And they're really interesting things. Some of them are relatively new to science and are not well known or characterized. This here is, again, reminder, this is at 40 millimeters below ground surface, which is much, much higher than repository depth, which we would predict to have a much lower abundance community and potentially much lower diversity community. But for now, this is the data that we have preliminary and available to show. Among those really interesting new to science taxa that we see, here are these nanoarchaeota and Pitesci bacteria that are a relatively high abundance in these samples, like the Woese archaea here and the Pace archaeota archaea here. So they add up, and they end up being among the dominant organisms in the subsurface. What these are, they tend to be these very, very small cells. The nanoarchaeota in particular has been known for many years from this Carl Stetter photo. They tend to be really small cells that grow only in association with larger normal bacteria and archaea. And yet there's a lot of them out there in the environment, and they have reduced sizes, reduced genomes, and they have a variety of different pieces of metabolisms that they encode. And a few years ago, Gillian Banfield's group published some groundwater data showing these purple bars here and a variety of these relatively shallow depth groundwater samples, that there are a lot of DPANN and candidate phylum radiation. These are these small things that we're finding as well. So our results seem to be consistent with others that are finding these really small, enigmatic, uncultured microorganisms that can contribute a variety of potential metabolisms, but including the potential for sulfur cycling, which is partly why they're of interest and why our sampling protocol has now changed to always include 0.1 micron filters in addition to the standard 0.2 micron filters, because the small things pass right through the 0.22 micron filters. So now we can characterize the complete microbial communities of the subsurface site, which I think is really important to do. All of this said, what's ongoing is more comprehensive site characterization that will be continuing beginning this year, right through for the next few years. And we are also analyzing pressure vessels, bentonite clay with metal coupons within them, placed at the site for different amounts of time. And you can see them here working with a pressure vessel that is characterized by our lab to look at a simulation of a repository with bentonite placed in the actual subsurface. So not just simulated groundwater, but actual groundwater. And this is a collaboration with Jamie Noel's group at Western University and ongoing research that I hope to be able to present on relatively soon. So lots of research ongoing, and I hope that I've managed to convince you through this presentation that when we sample different clays, we end up with different clay microbial profiles. And yet, despite that, we end up with very similar cultured bacteria out of each of them. So there are some predictable elements of what grows. And we do have good overlap between what grows and we detect on plates and what we detect with our ribosomal RNA profiles. And we see the bacteria increase in wetted clays, whether it's growth of the bacteria or animation from a dehydrated state is something that ongoing research will help to confirm. But it does appear that bacteria can grow in wetted clays, and this is primarily in the presence of oxygen and without pressure. A deep geological repository is anticipated to go anoxic relatively quickly, so that growth may be very transient and temporary and limited to that initial wetting phase only. And it's the pressure that leads to prevention of microbial growth, but even that very initial period of time where the deep geological repository is getting wet, that may be associated with a temporary increase in bacterial abundance, which needs to be considered and included in models for the microbiology of a DGR. But at the same time, we never saw sulfate-reducing bacteria growing during that saturation phase, at least in the data that we're presenting here today. So SRB growth not observed within bentonite. And then switching to the natural barrier components, we see undetectable microorganisms in rock core samples, so that's below our detection limits. And this subsurface water and vessel analyses have showed you some preliminary data at 40 meters below ground surface, but we have a lot more work to do to characterize the site that's been selected for a deep geological repository in Canada. And as mentioned, there are implications for other countries also considering a DGR, and so we continue to collaborate and communicate with others who are exploring similar questions in different contexts and geological formations, for example. So I hope that the data convince you that so far the microbiology results support the proposed natural and engineered barrier components for a Canadian deep geological repository, and we continue work in this direction to increase the confidence that we have of the microbiological compatibility of a deep geological repository. This work has many collaborators and partners at the Nuclear Waste Management Organization that I want to acknowledge. So the universities that we partner with is the University of Western Ontario, McMaster University, University of Toronto. And I also want to acknowledge the many people in my lab that have been contributing to this work over the past number of years. And I want to thank you as well for listening, for your attention, and I'm really grateful for the opportunity to present to you today. Thank you.